The overall goal of this protocol is to label individual transcripts in E. coli with fluorescently labeled DNA probes for visualization in single-molecule FISH experiments.
This method can help answer key questions in the systems biology fields, such as characterizing transcript cell-to-cell variability of gene of interest, as well as the transcript subcellular localization. The main advantage of this technique is that it provides quantitative information about the statistics of transcriptional processes.
To begin the protocol, grow E. coli MG1655 and LB medium overnight in an orbital shaker at 260 RPM and 37 degrees Celsius. The next day, dilute the overnight culture at 1 to 100 in fresh medium and measure its optical density in a spectrophotometer up to a value of 0.2 to 0.4. Next, pellet the cells by centrifugation for 5 minutes at 4,500 G at 4 degrees Celsius. Remove the supernatant from all tubes. Add 1 milliliter of 1x PBS to each tube, and resuspend the pellet to minimize cell-to-cell aggregation. Then add 4 milliliters of fixation buffer and gently vortex the tube. Incubate the samples in an orbital shaker.
Resuspend the pellet in order to minimize cell-to-cell aggregation, which may interfere with cell imaging.
After incubation,